we're reading this paper, Interneurons and Neocortical Inhibitory System. I thought I would start with a little background material here. Switch to my PowerPoint here. So there was a time in the past when I started studying neuroscience, there was two classifications of neurons. There were excitatory neurons and inhibitory neurons. And in the excitatory neurons, it was very clear what there were two types. There were pyramidal cells and spiny stellate cells. And there's no question about these cells. Everyone agrees they're there. They're very consistent. And then the inhibitory neurons are kind of a mixed bag. The reason the two pyramidal cells and spiny stellate cells were in the same category is they both had an excitatory effect on the postsynaptic cells. So if they would form a synapse on another cell, that would excite the other cell. Those synapses are all spiny, meaning they have the spines. They would only form these synapses on the dendrites. That's the only place they would form synapses. And it's generally assumed that this is where learning occurs, on these spiny synapses. So whether they're changing their effectiveness or they're growing new ones, that's where learning occurred. The inhibitory neurons. Yeah.
Clarify, sorry, just real quick, what the spiny refers to again?
It's that spine.
Because there's like the bouton in a synapse.
Well, a bouton is a fancy word for a spine.
Okay.
So this is a little character drawing here, but those are the spines. So the excitatory synapses are almost all on spines, meaning the axon and the dendrite don't actually connect to each other. There's a little spine that goes between them that grows out of the dendrite. So when the original people like Cajal would look at these images, they'd see these spines, they see these little bumps all over the dendrites. And they called those, those are spiny neurons or spiny synapses. Is that clear?
Yeah, thanks.
Yeah. Oops, I'm gonna go back here. No, I was in the PowerPoint again. This is contrasted in inhibitory neurons. When they form this, there's different types. There's lots of different types: large basket cells, small basket cells, chandelier cells, double bouquet cells, Martinati cells. Almost all these descriptions come back from the time when people are just looking at images and they're trying to describe what the cells look like. This is back 1900. They had no idea what any of this stuff was doing, but they would just say, hey, this is what it looks like. A pyramidal cell, they call it pyramidal because the cell body is like a triangle, looks like a little pyramid. And the spiny stellate cell is like a little star. And they wanted to distinguish from other stellate-looking cells. So they said, this is the one that has spiny synapses. Anyway, so there's all these different, and these other cells, these large baskets, these inhibitory neurons, they all have an inhibitory effect. They form a synapse, which makes the recipient cell less likely to fire. But they're inconsistent classifications and inconsistent nomenclature. So various researchers use different words for the same cells. It's kind of a mess. Those synapses are inhibitory and they're called aspiny. If you actually looked under my electron microscope, you would not see a spine. You'd see that there would just be a sort of a slight little thicker spot on the dendrite where the axon and the dendrite connect. The inhibitory synapses, they can form on the soma, the cell body. They can form on the axon hillock, which is the beginning of the axon, or they can form on the dendrite. So unlike the excitatory neurons, which only form, excitatory synapses are only formed on the dendrites, these inhibitory ones can be anywhere on the cell, but these three basic areas. And it's generally assumed that they're involved in regulation, not memory. So they would enforce sparsity or synchronization or rhythms or some sort of operational states of the system. That's the general view that people had and still have mostly. Let's talk about the pyramidal neurons, and only pyramidal neurons have apical dendrites. These are the big dendrites that go up towards the top of the layer of the cortex. And pyramidal cells, and only pyramidal cells, and all of them send a branch of their axon outside of the column. So all cells form lots of synapses locally within their column. but the pyramidal neurons always, there's no exceptions to this I'm aware of, will send an axon that leaves the cortex and goes someplace else. Stellate cells, the spinate stellate cells, look just like the pyramidal cells, but they don't have the apical dendrite and they don't have the long-range axon. So they form similar kind of local connections. And so there were some papers published, or at least one paper published that we read, that was basically arguing that stellate cells are really just pyramidal cells that don't have an apical dendrite. But otherwise they operate the same. Anyway, somewhere along the way, it was found out that some of the inhibitory neurons are not always inhibitory, and they can act in an excitatory fashion under some conditions. So they changed the nomenclature. So the new nomenclature is we have pyramidal neurons and interneurons. And interneurons are defined as neurons that don't send their axon outside of the local column. So they've moved the spinate stellate cell down into the interneuron category, which is very confusing because they're really not like these other neurons. They're really more like the pyramidal neurons, but this is the nomenclature that they've settled on, which can be confusing perhaps, maybe not, maybe it is, but it shouldn't bother me at a start because you can even see it in the paper we're about to review here. I just wanted to show you this picture just to make sure we're all on the same level when I'm talking about various things here. So this is a pyramidal neuron. This picture looks like it's a bit of complex. Part of it looks like it was actually a photomicrograph, and part of it looks like it was hand drawn. So I don't know. But here's a pyramidal cell, and it has this triangular cell body, which is why it's called a pyramidal. These are the basal dendrites here and here. There's lots of synapses on them. This is the apical dendrite here, and this is called the tuft. It's the upper part. Most pyramidal neurons don't have these little offshoots coming off, which are called Schaefer collaterals. Only some do, but most don't. This is the axon hillock here. You can see my pointer, I hope, right? You can see my pointer? Is that right?
Yeah.
Okay. So this is the axon, which also branches and profusely connects to other cells nearby. They're not showing that at all. But when we talk about the axon hillock, it's this very initial segment of the axon. So when a cell spikes, the body gets depolarized. The soma gets depolarized. And meaning it's normally at a resting potential of, let's say, minus 70 millivolts. It's polarized. To be depolarized, the voltage goes up a bit. And if it gets above a certain threshold, then the cell spikes. But what actually happens is the depolarization occurs here, but the action potential starts right here at the axon hillock. So there's these chandelier cells which form synapses right on this axon hillock, which will have a very big effect on whether this cell, actually sends a spike down its axon. Notice that this is not mentioned in the paper we're going to view here, but I've always wondered why would it do this, and we have some new data about it. But one thing, it probably wouldn't prevent the cell from sending a back action potential up its down its dendrites, because it's really just stopping the beginning of this action potential going down here. So there are synapses on the cell body. You can see some of the little bumps here, but those are all inhibitory synapses. You never find excitatory synapses on the cell body. You only find excitatory synapses along the dendrites out here. So there's inhibitory synapses on the axon hillock, on the cell body, and there's also inhibitory synapses, some spread out along here on these dendrites. It's a small percentage of the synapses, but there are definitely synapses along here, and so inhibitory synapses. So you'd imagine what's going on here is that these different types of inhibition have different effects. The ones I've always imagined, the ones that are along the dendrites, would probably just change the threshold for generating a dendritic spike. So they would make, if not enough cells were active, you might, if you change the inhibition out here, you could get these cells to generate more NMDA spikes or more dendritic spikes. Where the inhibition on the soma would effectively shut down all activity of the cell, and so we would imagine that would be good for creating sparsity. And the inhibitory synapses could do various things. One is that I just read, I didn't realize before, was that on the axon hillock, these synapses might be involved in getting cells to be synchronized, which is interesting. Okay, so so they show here, my cursor keeps disappearing. This is a chandelier cell which forms this little row of synapses here and lots of other pyramidal cells. It's called the chandelier cells because when they stained it, these look like little candles on a chandelier. And then they show here a double bouquet cell, which we've been talking about, which has this very narrow axon. This is the axon here, believe it or not. And it's this huge bushy string of axon that it seems to be associated with mini columns. And then here's a picture of a large basket cell, which just inhibits a lot of other cells, primarily by inhibiting the somas. So this gives you a sense of the kind of things we're talking about. Stop me if you want me to talk about something. All right, so here's the paper that Rami found and I read earlier and I agreed to review. The title itself is interesting because it says interneurons of the neocortical inhibitory system. So they're making the distinction. They're gonna leave out spiny stellate cells because they're only going to talk about inhibitory cells. So they already have this already. They're dealing with the fact that interneurons is not a good term for inhibitory cells anymore. So, and then this highlighted line at the end of the abstract says this review focuses on organizing principles that govern the diversity of inhibitory interneurons and their circuits. Again, they'll be mostly leaving out the spiny stellate cells, even though those are interneurons, because they're not inhibitory. It's essentially it's when most people have absolutely no idea of what the circuits in the cortex are doing. We have lots of ideas on that, but most people don't. And so what a lot of these scientists will do is they'll just try to come up with ways of characterizing cells. And this paper goes through a lot of different ways. You can look at these inhibitory interneurons and classify them in various different ways. It's quite complex. And a lot of it's not very useful. At least I don't find it useful. But that's what this paper is doing. It says, okay, we're going to talk about all these inhibitory interneurons and all the different ways we can organize them and classify them and say what they do and don't do. This line here, this gets a standard statement you'll see everywhere that in the neocortex, 70 to 80% of the cells are excitatory pyramidal neurons, also spiny stellate cells, which are smaller in number. And they have relatively stereotyped anatomical, physiological, and molecular properties. As I said earlier, the pyramidal cells are very, very similar. The remaining 20 to 30% of neocortical neurons are interneurons, mostly inhibitory, which have diverse morphological, physiological, molecular, and synaptic characteristics. So they're really different all over the map. And that's what they're going to try to talk about here. This little highlighted text here says interneurons can also be excitatory. This spiny stellate cell is an important type. So they're saying, yes, this is another interneuron, but for the most part, they don't really talk about it in the rest of the paper because it's not inhibitory. Here they say these cells are found only in layer 4. I was surprised in primary sensory meters. I know that was the way they originally thought of it, but I think they're found elsewhere now. So I don't know if that's still true anymore. And they also say, this is something I never saw before, they said the spiny stellate cells have a partial apical-like dendrite that can reach layer 3. I've never heard of that. I know they have their dendrites go spread around, but I never heard they have a partial apical-like dendrite. And I didn't see a reference for that, so I don't know why they said that. That's new. All right, here's the first figure. This figure is a very cartoonish drawing of the different types of cells, and it's talking about the anatomical diversity. So the cells actually don't look like this at all, of course. They threw up here the spiny stellate cell. That's an excitatory cell. Then they said there's some cells that target the soma and the proximal dendrites. So these will form inhibitory synapses on the cell body and the proximal dendrites. And in this category are mostly basket cells. I've often heard of large basket cells and small basket cells. I've never heard of nest basket cells, and they introduced that one here, so I don't know when that appeared. It's clear other scientists probably won't include the nest basket cell. Maybe they'll say it's a large or a small. So, how they classify these things is kind of it's somewhat arbitrary, not completely. Then they say these are cells that just target the dendrites, and then this category is the double bouquet cell, bipolar cells, which are also double bouquet cells are also bipolar cells, or at least some people say they are. And then there's a special here, this Martinetti cell, which is interesting because it seems to target primarily the layer 1 apical dendrites, and it spreads very wide in layer one. So we know that there's a lot of spread of activity in layer one as part of the paper we're writing. This is an inhibitory cell that seems to inhibit the dendrites in layer one. And then there's the chandelier cell we were just talking about, which forms these little cassettes of multiple synapses on the axon hillock and on pyramidal cells. So they're just giving us sort of the lay of the land here before they go into details. Any questions on that?
So we expect the soma and the proximal dendrite targeting cells to have a higher inhibitory effect than the other dendrite targeting cells.
Well, it has a different effect. One would, if you inhibit the soma, you're basically you're preventing the cell from firing at all, right? If I have an inhibitory synapse on a dendrite, I'm going to reduce the chance that it generates a dendritic spike, which is part of our theory is that the dendritic spikes are like a prediction. It doesn't prevent the cell from firing at all. It just says I'm going to make it harder or easier for the dendrite to generate a dendritic spike.
Right.
I've always imagined like, there's a threshold on dendrites, and like if you have like 15 to 20 synapses active in a very small area, we've written about this, 40 microns or so, it'll generate, and they become active at the same time, it'll generate a dendritic spike. Well, I just imagine, I don't have any evidence of this, but imagine if I have interspersed along that dendrite, I have some inhibitory synapses that are either more or less active. I can change the threshold from being 15 to 20 to maybe 20 to 25 or maybe 10 to 15. So one thing you could do is if you have too many cells that are becoming active, this could be, or if let's say there was not enough predictive activity, you could change the threshold and now you loosen things up. I used to always imagine, like if I say, oh, look at a cloud, you see the dog in the cloud. Well, there really isn't a dog in that cloud, and normally you wouldn't see a dog, but you do something in your brain, all of a sudden you say, oh yeah, I see there's a dog, the cloud looks like a dog. I always imagine what was going on there was that you were changing the thresholds on the dendrites. So you didn't have enough real evidence for a dog, but if you just lower the threshold a bit, you say, okay, maybe there's a dog.
Yeah.
Something like that. I'm not going to go through most of this paper because it's really, yeah.
Question I just had was, and maybe you're going to get to this, but when we talk about double bouquet cells, I feel like we often, at least computationally, kind of talk about them as excitatory neurons, or at least that's how I pictured it, in terms of like defining the receptive field. But is it something about like a inhibition of inhibition or something, or like how do they?
Yeah, right. I've generally kind of learned to ignore those ideas because what you might find, I didn't check this. The double bouquet cells a couple things may be happening. They may have a background firing rate, which then is reduced, and so they're at some constant threshold of inhibition. Then you reduce the inhibition, or there could be a double inhibition going on. Like, do these guys also innervate, basket
You like the chandelier cells?
You're right. It's complicated. It's very difficult to I found it difficult to track this down, and these kind of things. And so I used to worry about that a lot, Niels, and I just stopped worrying about it because it seems like.
Okay, yeah, no worries. That makes sense. Thanks.
Is the data there? Do we really know the background firing rates of double bouquet cells and what's it when the animal's attending versus not attending? It's like, oh my God, there's so many variables here that change in real life scenarios that it's just hard to get the data you would like to have for some of these things. This is another picture. They're just showing the propensity of the different cells in different layers. You could try to make something of this, but there's so much data here. You just can't look at this and go, oh, yeah, I see what's going on. It's just data. Right? And once you have a theory, you can sometimes go and look at data like this. Say, oh, maybe this supports or doesn't support the theory. But up front, it just says, okay, in layer 6, this is how many of these cells you have. It might be interesting. Okay, they got more small basket cells than any other layer. Okay, well, the general idea, I think, for a small basket cells, it's this basket cells, which I believe, and other people suggested, are just generally there to create sparsity, so you make sure not too many cells are firing. And the small ones are more limited in area, so it might be within a column. And the large ones seem to extend beyond the column, so that would be like column to column inhibition as well. So anyway, the small basket cells thing, maybe you have higher sparsity down here. I don't know. I'm not going to go through each cell here. This is the one here on the double bouquet cells, since we've been talking about them a lot. These words all describe what we've been talking about. Have a bitufted, meaning it goes in both directions, up and down. Morphology for the dendrites, not the axons. We're talking about the dendritic morphology. Their special feature is a tight axonal cylinder. So the thing that's really tight and that's narrow is the axon. And I don't even know what fascicular means. I think it means, I don't know, highly branching or something like that. And then resembles a horsetail. Sometimes they actually call these horsetail cells. So you might see that in early literature. They talk about some of the details of it. And it says they're interleaved with pyramidal cells, inhibit their basal dendrites. See here. Okay. Anyway, this is just language to describe what the cells look like. Then we get into things like this. There's a whole bunch of there's neuropeptides and there's proteins that they can use to categorize these cells. I'm not even sure exactly how they categorize them, but then you can so that they're listing some three these calcium binding proteins here, and then these neuropeptides that are associated with different cell types. And then on the right is different types of firing patterns. They can characterize what the spike patterns look like. Adapting with A here, non-adapting here, stuttering, and so on. I forget what IS stands for. Irregular spiking. So they make up names for these different patterns they see and say, well, I'm going to call it that. And then you can associate the cell with the neuropeptide, with the different patterns in which they spike. And okay, yeah, I have no idea what to do with that. You know, it's like it's there, but it's like, what are you going to do with it? This was an interesting one up here, the highlighted text. So we're talking about electrical properties of neurons. Originally, all inhibitory neurons were described as fast spiking. So what that means is, it could be two things. One is how quickly they generate the first spike, but also then how fast do the spikes occur. But I think the more important one is how quickly they generate the first spike. And so our theory of the temporal memory theory requires that when a pyramidal cell becomes active, it then inhibits all of its neighbors. And so the pyramidal cell activates an inhibitory neuron. The inhibitory then has to activate all the other cells before they can spike. And this system has to work really fast. And someone did the simulation of this on our behalf, and they said, yeah, it could work. So that's an important part of our theory that happened. Now they talk about here in the paper, they talk about the different response properties of these cells. Unfortunately, these response properties are almost always detected in some sort of slice preparation. So they've taken the brain out of the animal, they sliced it up, they put in some liquid to keep it sort of alive. And then there's two different ways they do it. There's a steady state response types, meaning they inject current into the soma, into the cell body. So they just a little electrical probe, and they just inject current continuously, sustained current, and so the cell doesn't get to relax. And so then they say, oh, well, what kind of spikes does it generate? And then the other one is they can do a step depolarization, like what happens on the very initial pulse of current. So onset response types, and so they can classify those. Before I go on, this is another figure they've thrown in here, which is showing the number of synapses that individuals, interneurons, make onto pyramidal cells. So, you know, there's varying numbers. Again, very hard to interpret in any meaningful way. Then they say, you know, okay, we're looking at these response types, you know, like a pulse. Well, how's it respond to a pulse, and how's it respond to current injection? And then they say, well, you know, it's a useful means, but maybe they're not really real. So they say extrapolating these, here I said, what I say here, slice preparation may be misleading. That's my summary of this. Extrapolating these types of in vivo simulation, meaning to the in vivo, like what would actually happen in a live animal, is really it's hard to say, because the brain is sliced, perfused with artificial fluid, and we see this greater synaptic environment and are subject to neuromodulator control, which is which could profoundly change its discharge property. So basically, it's saying, yeah, these things, the way we characterize these cells may not be accurate at all in a live animal. So it's not that it's not useful, but you have to just remember that these things aren't like set in stone. Then they talked about ion channel composition. So ion channels, these are the channels along the cell membrane of a cell, and they can parse these interneurons by different types of ion channels. Again, not really easy to interpret in any revealing way. And then they can look at genetic expressions, and they can go all kinds of crazy stuff. Gene clusters. It's not really worth going into all this. These are some of the different these are different cell response properties, and this is based on, you can see down my highlight here, based on steady state response to sustained current injection in the soma. Again, they have a cell in slice, they inject current into the cell, and some cells they'll behave different ways, and they'll have like a series of spikes or non-series of spikes or a little burst in between, and they put names on these things, trying to sort these into classifications, which are not unreasonable, but again, very, very difficult to interpret, to say, well, what's the point of this, right? I think one thing we've learned, and then this is something that Sherman and Gilly have really proposed a lot, is that most pyramidal neurons and most stellate cells, they generally have they don't really burst very often. They have a steady state spiking. And when you have a little burst, like these things here are bursts, these things here are bursts, or even at the beginning of this one, here's a little burst of a bunch of spikes right in a row really quickly. When this reaches the destination neuron, those bursts can cause metabolic changes, meaning they can cause the receiving cell to actually grow something or change something physically. It can express genes and make things happen that are not just like a voltage depolarization. So these bursts really do matter, but it's very complex. It's hard to say exactly. I'm not going to go through most of this stuff here. Here's another take of the same things, different cell types, different types of spiking patterns. I'm not actually sure. This is according to discharge responses at steady state and onset phase. So this is like I think this is like the pulse version of the one I showed earlier. Again, you look at this and go, what am I going to do with this information? It's really hard to know what to do. So here is they talk about the interneurons themselves, even though they're mostly inhibitory, they get excitatory synapses. So they talk a bit about that. Let's see. I'm not going to skip down this and want to talk about this stuff. There's some interesting clues here. Like they're talking about some sort of delays and stuff, and they say which might cause targeted inhibitory cells deep in cortical discharge before those in supragranular layers. So this is the kind of thing I would pick up and say, oh, that's interesting. They're basically saying that there's some properties here which might make the upper layers go first and the lower layers go second, or actually the opposite way. The lower layers go first, the upper layers go second. So I just highlight things that catch my attention like that. This is another lovely but really not very useful chart. It shows the let's see, correlation map between the different ion channel genes with specific electrical parameters. So I guess if you were like studying some disease and there was some disease related with various ion channels and you want to know the genes that are expressing to make those ion channels, this might be useful. But from my point of view, trying to understand how the brain works, it's not very useful at all. Nothing bad about it, just can't really do anything with it. This is this is interesting here. So there's this new idea which I'd read once, but I just got more introduced to it very recently, is that these inhibitory cells aren't always inhibitory. The same synapses can be inhibitory or excitatory under different conditions. So it says here, a single neocortical neuron can receive both depressing and facilitated GABA in synapses. GABA is the inhibitory neurotransmitter. If an interneuron has a delayed onset response, synapses tend to be depressing. If the onset response is classic or bursting, synapses tend to be facilitating. So they're saying, okay, we now are saying some of these synapses can be inhibitory, some can be excitatory. Let's see here. Look at this orange one here. I said neurons that preferentially target an axon initial segments. Those are the chandelier cells. And I'm going to skip the rest of the important part of the sentences. Are involved in phasing and synchronizing neural activity. So this is really interesting. I used to think that, like all these inhibitory synapses would just prevent a cell from firing. But that may be true, but misleading, because it really may be just saying, don't fire right this second. I want you to wait a little bit or fire a little bit earlier. And it might be just saying, I want you to change the timing of your spike as opposed to spike or non-spike. Let me just jump to another paper here, which I have. This is a paper about chandelier cells. And these are the ones that form the synapses on the axon hillock. And I read this paper. Here's like a diagram. Here's the red is their axons. These are little things that are on each pyramidal cell's axon hillock. And then here's here. Okay, so this sentence here says, activation of the chandelier cells shortly before the expected pyramidal neuron spike evoked reliable inhibition. On the other hand, activation of the chandelier cell at earlier points in time evoked hyperpolarization and depolarizing effects. They're saying is if the chandelier cell fires right before the pyramidal cell is about to fire, it will inhibit the pyramidal cell. But other times it might excite or make the cell more likely to fire. And so the idea here is it's similar to like timing-dependent plasticity. This seems to be like a timing-dependent spike formation. And it suggests that the chandelier cells may be actually just, they may be just changing when the pyramidal cell actually generates a spike. And it would be a way for getting pyramidal cells to all spike at the same time. This has always been an odd thing in neuroscience, because for like dendritic spikes to occur, these action potentials have to arrive at pretty much the same time, within a millisecond or a few milliseconds. And so this is a really interesting idea that these chandelier cells may be really changing the spike timing exactly of when spikes come out of the pyramidal cell. And it may be a synchronization method. And I've seen some other references to that as well. So that's pretty cool. So that's what they're referring to here. Neurons that preferentially target initial segments, which would be the chandelier cells, are involved in phasing and synchronizing neural activity. And there's some references there which I didn't read. So that would be, if I went on to follow that idea, you should look at those three references. Here's another chart that's mind-numbing. Mapping glutamate synapses from neocortical pyramidal neurons onto interneurons. So these are like, here's a pyramidal cell, and they're saying, okay, the pyramidal cell is going to form excitatory synapses on these different types of inhibitory cells, and what kind of synaptic responses you get on those inhibitory cells. And so yeah, that's what that chart's about. Again, I don't know what to do with that. Let's keep going down here. This is interesting here. They say, why balance yin with yang with their yang with yin? What they're talking about here is the neurons want to be excited, but then you have this inhibition of basically saying, why do you have excitatory and inhibitory things going on at the same time? And there's no mention in this paper at all about sparsity and advantages of sparsity or why you'd want to inhibit neurons. I don't recall them ever saying anywhere in this paper why you'd want to inhibit neurons. But I thought that's pretty funny that they don't go there. Again, they talk about, there's different ways of classifying the things. Maybe we're not getting it right at all. I just highlighted this section because it had a lot of good statistics. Not that we need to go through them, but it had a lot of numbers. About 60% of the synapses on a pyramidal neuron in layers 2, 3 are inhibitory. And these come from about 70 interneurons, half being basket cells. Roughly 50 of these are neurons from the same layer, blah, blah, blah. So this is the kind of thing like you'd say, well, someday if I ever really needed to get some details on this, there was a nice paragraph on that. And then they conclude, and the conclusion is just a summary of there's a common cortical circuit. A template neocortical microcircuit seems to have been duplicated repeatedly to construct a neocortex. In terms of its general architecture, this template is highly stereotypical, quote, a generic microcircuit with subtle specializations that presumably optimize processing in different brain regions. In terms of its functions, the generic microcircuit at any one location seems to support simultaneous processing of multiple features. I don't know what they meant by that. Anyway, basically they're saying there's a common microcircuit here. All right, so that's my read on this paper. There hasn't been much discussion, but I don't know if you have any thoughts or questions about it.
Really interesting.
I have maybe one question. Somewhere in the paper, they talk about something called differential synaptic transmission. And
what was the word? I didn't hear the word.
Differential synaptic. Let me maybe share my screen.
Okay. That's your own notes. Your notes are probably better than my notes.
Differential. Yeah, they talk about something called differential synaptic transmission. And can you see my screen?
Yeah.
And the pointer. And yeah, they refer to it as some sort of short-term synaptic plasticity, where the way I interpreted it is from excitatory cells that they can basically, these excitatory cells can kind of like route their neurotransmitters, or they can cause some dendrites to be more depressed or facilitated in a short term that usually lasts less than a second. When I looked up that synaptic plasticity thing, and the way I interpret it is that these excitatory can actually cause different dendrites to have different effects on the postsynaptic neuron. And I really didn't know that before. I don't know if this is true or not. I was just checking.
Well, I'm still not sure what it means. Can you scroll down so I can see the sentence before this?
Oh.
What are we looking at in the paper here?
So here they're talking.
We're in the category of excitatory synapses on interneurons. Is that where we are?
Yeah.
Okay, and then all right, they're saying these synapses typically form in clusters, a small fraction of the dendrites. Okay, so these are excitatory synapses on inhibitory cells, I think, right? And right. And instead of being spread out like we require on pyramidal cells, because we're trying to recognize sparse patterns, here they're just grouped together, meaning like this excitatory cell is just going to activate this inhibitory cell. Okay, so that's where we are. I'm trying to work my way up to where you had that sentence, which I didn't understand. Physiological properties. Okay, so unlike glutamatergic synapses on excitatory cells, glutamatergic synapses on inhibitory cells are different. Okay, can you go back to the top of the page now? Okay. And often have frequency-dependent facilitation. These features often found in some interneurons. some different types of physical behavior. The glutamatergic system may have a functional dichotomy of different modes for recruiting pyramidal neurons and interneurons.
Yeah,
I feel like
then-
it's actually clearer if you scroll down the paragraph under.
yeah.
I feel like that kind of, it's a weird ordering because I feel like this kind of explains it. It's basically like synapses from excitatory cells on inhibitory neurons that actually has an inhibitory effect, is my understanding.
The static dynamic depression and facilitation vary across layers. Which may cause target inhibitory cells and
Like they're basically depressing synapses even though they're coming
Yeah.
from excitatory
Yeah.
cells, which is why I guess they're called differential, because it's like different from normally what
normal.
glutamate does.
Right. At first I'm thinking like differential, like there's some difference in time, but they're basically just saying, hey, the same cell can have different effects.
Right, the different effects
Okay.
on different dendrites.
So okay, so let's figure one time to figure that out. That goes back to, I guess, the earlier comment I made earlier, where they used to think these things were just pure inhibitory. You know, everyone assumed that, you know, the same kind of synapses result in the same kind of things, but it's not always the case. So what was it? Now, are you confused by this? What's the
No, it just the way I interpret it is that the same excitatory cell, when it releases when it spikes or makes an action potential, it doesn't have the same effect on all of the synapses. It can actually either facilitate or depress some of those synapses.
But those are just synapses on inhibitory neurons, right?
Yeah.
I don't think that's true on when a pyramidal cell connects to a pyramidal cell. I don't think that's true.
Yeah, that was part of what I was wanted
Yeah.
to.
So then I would put this into, you know, I made this bucket early on, like, is this where learning occurs, or is this some sort of regulatory function, right? And this, to me, as soon as you're forming synapses on these inhibitory cells or the interneurons, you're into some sort of regulatory function. And once you're into the regulatory function, it's a wild zoo of unknown possibilities, right? Anything could happen, and it almost seems like anything does happen, right? It's a really different story. So, I guess that kind of stuff, Rami, that is like one of 20 things in this paper that are like, oh, that's interesting, okay, what am I supposed to make of that? So again, my take on this kind of stuff is, you know, we started off by just totally ignoring inhibitory cells in our theories, other than we said we have to have sparsity. And because there's just too much unknown, there's too many weird stuff, and it doesn't seem to be where learning is occurring. So then we just focus on how could learning occur within excitatory cells, pyramidal cells, you know, and then when we need a regulatory function, well, you know, we can go look into the bag of, you know, mixed mess stuff like in this paper and try to find it. Like, you know, how do grid cells work, right? Well, that requires all kinds of weird stuff. And some of it appears to require sort of shift in, you know, phases of certain cells. And, you know, that's probably going to be happening using interneurons with some sort of, I don't know, who the hell knows how that happens? No one knows. So you're kind of like, okay, well, if we need something, if you need a system to do something, it's probably going to be done by these interneurons, and maybe we can tease out how they do it. The example I gave earlier, where the basket cell had to be had to support this idea that we have, you know, a burst of cells, then a quickly winning cell in the temporal memory algorithm. Well, that was the first time we could say, okay, we need an inhibitory cell to do something specific. And the basket cells, we're pretty sure that's them, and they're pretty well characterized. And so now we had a reason to go look at basket cells and see, could they do what we think they need to do? And that was a positive experience. Whereas most of these cells, we don't have anything like that at all.
Have we considered that perhaps maybe the double bouquet cells are doing the inhibition across the column, like for lateral inhibition in one column?
A lateral inhibition in a column?
Yeah, like if one of the cells in a mini column excites that and hit the double bouquet cell.
Well, it could be. Oh no, I don't think so. Actually, I think the double bouquet cells are slow, if I recall. They're not very fast cells, and they wouldn't fit that purpose. And they would, yeah, I don't think so. I think that's the main reason they're kind of slow. There was an interesting question I had early on, which is like, well, the classic Hubel and Wiesel experiments, they show all these cells in the minicom have the same basic response properties. And I always ask myself, well, how the hell did that happen? What makes these cells have the same response properties? And most neuroscientists just assumed it was genetic. And I always felt like, no, that can't be true because, and they would argue that, look, when an animal's born, it already has those response properties, but then there's these waves that go across the retina, and it's complicated. But I said throughout the cortex, it's almost impossible that all those mini-coms have the response properties are genetically determined, has to be learned. So what forces these cells to have the same response properties? So I actually, before I knew a lot about double bouquet cells, I said there has to be some sort of interneuron that is enforcing that. There just has to be some mechanism that forces these things and forces them to all have the same response property. And on top of that, that cell, and so then I looked at double bouquet cells, sort of fit the bill. They're just perfectly aligned with these minicoms. At least they have the ability to control all the cells in the minicom in some way. And then we also said, well, then those double bouquet cells probably also have to have the same response property as the cells in the minicom, and sure enough, they do. So that was another prediction that said that they should respond, and they do. The double bouquet cells do get input directly from LGN or the retina, from the thalamus, and so that all fit beautifully. So okay, well, they're going to be defining what the minicom is. They're like the spatial Pooler algorithm would be between these double bouquet cells. They would then somehow enforce all the cells in that minicom to have the same response property, albeit Neil's objection, which is a good one, like, hey, it seems to be inhibiting them. Well, maybe, I don't know. We could figure that out later. but physically, it's the only thing that fit, right? It's the only thing. Another interesting thing that came out, I just read recently, there seems to be just one double bouquet cell per minicom in the upper layer and the lower layer, and which is really interesting because that also makes it easier because just that one cell is going to determine the response property as well the 50 cells below it, or the 20 cells below, whatever it's doing. So these are the kind of ways that we work these things into the theory.
Yeah, one thing I found interesting in this paper is that they talk about inhibition of inhibition. There's a whole section about inhibitories,
right.
synapses on inhibitory. And they say somewhere that the basket cells are more interconnected than double bouquet cells. So the double bouquet cells don't really inhibit each other that much, but the basket cells do in layer 4.
I don't know what to make of that. I would think that I would want. There needs to be, if it's the spatial Pooler, there has to be some way that the double bouquet cells do not all become active at once. They have to have some sort of inhibition. Another thing to be careful of when they say that there's more inhibition here, more inhibition there, they may be just doing it by the numbers. They may be just looking at how many synapses there are, which is a terrible way of doing it because one strategically placed synapse can do everything, and 50 non-strategically placed synapses may do very little at all, or they may be encoding some pattern or something like that. So you'd have to look carefully at that kind of data. That's what I've learned.
Yeah, these cells are a lot more complex than I wanted them to be.
Right. That's why I just say, throw them into the bucket of regulatory functions, and then when we need something, we'll see if it's there to support our need.
Yeah.
This recently, I've had, based on this idea that's going on in the grid cell literature, that the idea occurred that the cells in a minicom all have the same response properties, but perhaps they all have different phases. And that's something that that grid cells require in some sense. And that I've never heard anyone write about that or talk about it or test it or anything, which is a really interesting idea, that these double bouquet cells may not only be assigning each of these cells the same response property, but literally the propagation delays or something like that along the double bouquet cell axon, which is a slow cell, might be enforcing the phase shift as you go along the cells in a minicom. So that's another idea that came up in the last couple of years.
Yeah, because these double bouquet cells, they get their input from also from the thalamus, just like
They do, but more importantly, you know, most of the cells in let's say in B1, if you look at the upper layers 2, 3, and 4, most of the cells are respond to movement. They're not, you know, the layer 4 has a lot of cells that respond, are more static, but layer 3 cells are primarily, or many of them are movement sensitive cells. And the theory we have there is that they're actually detecting, they're doing path integration of components of an object, you know, like it's behaviors of an object. And that's the theory I've been working on recently. And so then if that were true, if there were like another set of path integration neurons up in layer 3, like you're trying to feel like, okay, I'm trying to learn the movement of the top of the stapler. I need to detect its movement and know where it is. Then you would need these phase shifts up in the upper layers. And so maybe layer 3 cells will have phase shifts in them to do the which is required for grid cells and path integration. So a lot of unknowns there. It's very speculative, but you know, somehow this is all going to come together. And there's a situation. We know that the cortex has to learn behaviors of objects. We know that that's part of the object's model. We know that these cells are movement sensitive, so that's probably what they're doing. I can't imagine what else they're doing, that they're movement sensitive. We know that they're limited in their space, as the layer 3 cells detect movements over small areas. So it all fits together nicely.
Yeah. That was really a great review. Thanks, Jeff.
Well, thanks. You picked it up, and you wrote a lot of nice notes on it. I wouldn't have picked this paper to review other than you picked it. So it's okay. It was a good opportunity to, if you want to get up on neuroscience, you can.
Yeah, I feel like I'm missing a lot of the background, so I wanted to pick a paper that, you know.
Yeah, well, I thought it was a good way of doing sort of background neuroscience, and then I could talk about the basic ideas and what's happening and how the field has changed a bit and so on.
Yeah.
So I kind of enjoyed doing it.
Yeah, that was great. Thanks for suggesting that paper, Rami.
All right.
Yeah.
That's all I have on that.
And yeah, in the Slack channel, Rami, you posted some interesting notes about how that might relate to some of the theory discussions we had lately. I don't know if you want to go more into those or.
I have some notes, but I think we've covered a lot of them.
Yeah, we covered a lot of those, I think. Yes. Yeah. One.
I purposely didn't read your notes because I wanted to see. I just didn't want to be biased by them, and I want to see if my take was similar to your take, or if maybe.
Yeah, I also skipped a lot of the parts that they talk about ion stuff and all that. I skipped a lot of the things in the middle of the paper, and then I picked up like later section. I felt like this was too much detail that I didn't need.
It is, right. It's huge amounts of detail. And who knows if it's useful, but the tools I have, and they have these tools, and so they're going to use them. You know, if outside of any kind of theory about what's going on, this is the best that they can do.
Yeah.
So.
Yeah, I guess just one comment you had on Slack, Rami, which I'm not sure we really touched on, but was just that, yeah, I think during one of the brainstorming sessions, Jeff, you were speculating about double bouquet cells being not just responsible for kind of aligning receptive fields in layer 4, but also elsewhere. And I think Rami was just pointing out that, yeah, this paper supports that double bouquet cells are, I think, in like every layer except for L6.
And that they are in layer 2, 3, but also in layer 5 in large quantities.
Yeah, and then there was another paper I read recently, just recently, that said they only exist in layer 3. So you just can't believe this stuff, you know. Well, to me, it makes sense that you would be a set in the lower layers and you'd be a set in the upper layers. And you know, when they say, it's interesting, when they say they're only in layer 5, does that mean that their axons don't go to layer 6, or is that their cell bodies in layer 5?
Yeah, no, that's a great question. I'm not sure.
I would be a little disappointed if it didn't go into layer 6. Well, the idea that the double bouquet cells may be assigning this sort of phase shift that's required for grid cells, would be nice to see that down in layer 6. So just like, the double bouquet cells in layer 3 project, or 2 project to 3 and 4.
Yeah, they just say that they occur in layers 2 to 5, but given their nature of their axons, it seems reasonable to expect that they would project down to layer 6. But yeah,
reasonable, but probably somebody knows. Maybe it's probably written someplace.
Yeah.
Yeah, that's a good hypothesis. I mean, that's a prediction that we'd like to see that. If we didn't see it, I'd have to rethink that theory a bit. Like, okay, shit, it didn't work. Anyway, I think from the point of Monty, of course, none of this is terribly important. So, but it's helpful to be knowledgeable.
In the paper, somewhere they talk about late spiking cells, inhibitory cells. And I was wondering if we've incorporated any of those in the theories of how.
Is that just means that the initial spike doesn't occur, it takes a long time?
Yeah. So when they do a depolarization step, it takes a longer time to spike for the initial spike.
I thought double bouquet cells had that property. Is that not true?
They refer to. Let me bring it up quickly. I think it's something called NGC or.
Oh, those are the neurogliaform cells?
Yeah, they said.
I didn't talk about those.
Yeah, I think that they're in small quantities. I'm not sure. These
right.
ones.
There's these neurogliaform cells, which are like the hell they are. I have no idea. They're teeny little cells. They don't arterize much. There's very little literature on them.
Yeah, and they said that they exist in layers 2 and 3. Some in layer 5.
They're so. I'm not seeing anything. If you're projecting anything, I'm not seeing it. There are so few. There's so few literature about these neurogliaform cells.
Yeah.
And they have such a small spread of activity. I mean, they're really. I get the impression they're really small and they don't really extend very far. So it's like maybe they're doing something else. Maybe they're like cleanup functions, or who knows what they're doing. It could be like just doing some housekeeping. I don't know.
And they didn't talk about them much either in the paper. It's just.
I mean, if you want a cell to do and have an important role in the cortex, it's going to have to form a lot of synapses with other cells and have some sort of presence of like, okay, this thing is connecting to all the axon helix and whatever, right? As opposed to like, oh, there's little cells over here that just seem to be, sitting over there. I don't know. There's lots of other cells in the neocortex. There's tons and tons of non-neural cells. And some of them play important roles, but they're not general information processing roles. So,
just on the double bouquet cells, I just had a quick look at, of course, the Thompson paper, always useful. But I can just quickly share. But
I just keep hoping she's right, because we
yeah,
go
right.
back to so many times. It's like,
I mean, it's kind of like one of the only. Like anytime you Google anything about layer 6, this paper always comes up. But yeah,
right. So this is 1981. Look at the date on that. Right? Okay.
Yeah. The sources, but this paper is 2010.
I know, but she's citing a source from 1981.
Right. But yeah, just that layer 4 double bouquet cells at least project down to layer 6, and I think this is what you were talking about, that it's fine unmyelinated axons. So that would be potentially slow.
Very slow.
And then here it's also described as slower dendritic inhibition may come from layer 4 double bouquet cells to layer 6.
Yeah, so that's great. I maybe I knew that, maybe I didn't know it. I can't remember.
I think, when I looked at this, I remember us talking about this, and I think that's where the earlier discussion, like when you were saying about slow inhibition, I think that's where that came from.
Right. I mean, it can be really slow. So the information could. I mean, interesting. I wonder how fast we could look it up. How slow can an action potential travel on a thin? Thin makes it slow, and unmyelinated makes it slow. If that could, if it could take, a theta cycle to get, let's say 120 milliseconds to get from one end to the other. You know what I'm saying? That number exists someplace.
Yeah, there's this nice study, I'm just pulling it up, of it's actually from birds, but they looked at the conduction delays, and these were local connections. Oh, here we go. Yeah,
This is-
conduction delays, and
This is along an axon?
yeah, so yeah, axonal conduction delays measured from the peripheral to
Millimeters
hang
per
on, that's from the
second.
peripheral to the CNS. Intracortical axons unmyelinated.
Okay, let's take the number. It looks like I would say that this is a log scale, so that's like 0.2. Is that right? That blue one, intracortical axons unmyelinated. Let's just take
Yeah.
that one. 0.2 millimeters per second. So how
Per
far would
per millisecond.
millisecond? How far would I go? See, if I wanted to go, if I had per
So, I
millisecond.
mean, here they kind of do the calculation for you: distribution of axonal path lengths and resulting conduction delays. And basically, at the upper extreme, at the very upper extreme, and these are rare, is like 20 milliseconds. But you can see like a significant proportion go up to 7 milliseconds.
Well, this is to go from where to where?
This is on the scale of, I guess, half a millimeter, maybe.
All right, so that's not enough, really. I mean, if I was trying to get a disperse, if I was trying to get all phases of a
Yeah.
theta cycle, which is maybe 8 or 10 hertz, let's say, 100, you have to go 100, 100 milliseconds. So this is not, doesn't. work
Yeah,
out.
I think there's no evidence for that slow.
Yeah, okay. Well, it's interesting, those axons from the, I believe these axons from the double K cells go up and down, up and down. So maybe it's doing a circuit, it's building on itself. I mean, I wouldn't put it out of the question. It's not impossible. What would happen if you did something like that? You might have some interference pattern that, I mean, that's actually not a bad idea, actually. Imagine you have this action potential going down, takes, maybe it takes 10 milliseconds, and then it comes back up again. It goes down again. As those signals pass, they might be interfering, and you might get some sort of the interference pattern could be much longer than the delay.
Yeah, where you just have some intermediary neuron, and then it's like a recurrent circuit.
Or no,
that goes back and forth a few times. So like it's still synapses, but
I'm a pyramidal cell, and I'm getting multiple synapses from the same double bouquet cell, and they get quite a few. There's a number in there. So those synapses could be formed not from different branches of the axon passing by, and so when those actual synapses fire would be offset, and in some situations they might align, and you get, it's the same kind of oscillatory interference that you get with good cells. It might actually, the delay, I'd have to work it out, but I could see that there could be sort of a summation of these different delays that results in a much longer delay. I don't think about that.
right.
Something's going to make it happen. So you can almost guarantee that it's going to happen. So there has to be some neural mechanism to make it happen. That's interesting. Again, from our theory's point of view, it's really cool and interesting. From neuroscience, it's pretty cool and interesting. But from a Monty point of view, it's not that important to understand that stuff. That's interesting. That's a really interesting idea. That's a good chart. What paper was that in?
It's from this paper by Egger et al. It was a neuron paper from, I can show it on the group, from 2020, where what do you call it? They
were doing this for birdsong.
Yeah, and so they basically claim that the axonal conduction delay serves like an important purpose to the spatiotemporal sequences. But there's this big question. Okay, this is in birds, and it's in like a a part of their brain that's because it's what controls the pattern of their bird song. So
Right.
clearly time is very important.
But
And
there
so,
might be something. I mean, how a
but
bird
there
song,
could be something similar. Yeah.
bird songs aren't that short. I mean, so they have to have similar timing requirements that might last, 100 milliseconds or 200 milliseconds.
Yeah. Oh yeah, and actually, if I remember correctly, they also measure induction delays in rats.
I mean, if they're going to somehow explain bird song, they have to go from being something that's only a 10 millisecond delay to songs that, are longer than that. But
Oh yeah, that makes more sense. Because I was going to say HPC premotor neuron, L4 spiny neuron. That sounds like mammalian stuff, but who knows? But
Yeah.
anyways, yeah. So this is from rat somatosensory cortex.
What's HVC? I don't know what HVC is. HVC
Maybe the
premotor.
HVC was the bird part. Sorry, and then I think the somatosensory is the.
Well, that chart, interesting. You go back to that. So they have something there that's less than point. It's less than 0.2, right?
I would avoid looking at A because that's like a logarithmic scale.
I know.
just.
I know. Well, I'm reading off the logarithmic scale.
But I guess just there you go.
The path of the distance. Milliseconds.
I think what they're.
I'm trying to look. I'm trying to see a chart where it has the length and the delay, but I have the length and the frequency and delay and frequency. I'm not trying to.
So you have the delay here and the length and sorry, the length here and the delay here. I think the point they're maybe trying to make is that although the distribution of delay of lengths is different between the species, the delays tend to be the same and tend to be long. So I guess they're arguing that maybe that's like a conserved property or something.
Yeah.
That's important.
All right. Well, I don't know.
HBC is a part of the bird's brain that's important for producing the songs.
Okay.
Yeah.
Yeah. All right. Well, I mean, bird songs have durations of, they're not super short. So they have to have timing mechanisms that work for playing back sequences like that.
Yeah, and the paper is basically a combination of measuring these kinds of delays and then putting them in a spiky neural network model and showing that having these delays better explains the sequence produced than without.
So the birdsong mechanism is highly specific, right? And if I recall, unlike us listening to a melody, I can hear a melody faster and slower, and I still know it's the same melody. I think birds are not like that. I think if you just tweak it even the smallest amount, they don't recognize it. It's like really rigid. So they're probably using different mechanisms than we do. Well, anyway, all interesting stuff.
Oh, one more interesting statistics I found it just before we go. Yeah, these so when they're applying balanced inhibition, this is under the section of balancing yin with yang, and they talk about how the inhibitory synapses are different from excitatory, and they talk about how the time course of inhibitory synapses is about twofold longer than excitatory currents. They also talk about interneurons can discharge 2 to 3 times faster than pyramidal neurons. So I think this goes back to the idea of fast spiking that Jeff was.
Yeah. The time course of inhibitory synaptic currents. Well, I mean, if I want the inhibitory neuron to fire very quickly, what does that mean? The time course of inhibitory synaptic is longer.
Keeps sending neurotransmitters for faster, like for longer time until.
Oh, I see. It's not that it happens. It just stays active.
Yeah.
It stays active longer, right? Right.
They're trying to balance this with the idea that the 16% of all synapses on pyramidal neurons are inhibitory. So they're saying if they're only 16%, then how can we balance it with inhibitory synapses?
But that's a really crazy idea too, because this is ignoring the whole idea that dendrites are active processing elements, right? If you think about a dendritic tree as just like, oh, it's just summing the inputs from all these synapses, then you could say that. You could say like, well, there's not, we have a lot of excitatory synapses, not as many inhibitory synapses, so how can that be? But if you think about, that's based on a very, very poor thinking about how dendrites work. If you think about dendrites as being, these active integration zones and the threshold of local dendritic spikes, then this concern goes away completely. So and this paper was written, this is 2004, so it's 20 years ago. But maybe we can forgive them. I don't remember when people really started understanding dendritic spikes. Maybe it was around that time. I don't know. But nowadays you would never write that, because we know that dendrites are much more. They're not just summation plots.
Yeah, that makes sense.
Right.